Hello, my name is Emily Muff, and we're going to discuss Lysinuric protein intolerance. So, humans require 20 different types of amino acids, all of which are vital to their health and dietary needs. Not only is the consumption of these amino acids important, but the ability to digest and process them is equally as important. A patient diagnosed with Lysinuric protein intolerance is unable to digest 3 of the 20 amino acids. This is due to a defect within the small intestinal wall. While the condition is very rare, it affects males and females equally, skips generations, and has been found most often in patients of Italian and Finnish descent. It is important to note that consanguinity is present in some of the affected families, which will be discussed further detail later. Lysinuric protein intolerance presents itself after an infant is weaned off breast milk or formula, making the disease deadly in childhood. The reason why LPI does not present itself until after solid foods are introduced is due to the low protein concentration in breast milk. Lysine, arginine, and ornithine, which are the 3 amino acids out of the 20 that are not digestible in LPI patients, are found in protein-rich foods. After an individual consumes protein without the ability to digest it, they experience nausea and vomiting. Due to the body's lack of protein, it is common for these patients to be short in stature, experience muscle weakness, osteoporosis, end-stage renal disease, comas, and intellectual disabilities. Also, a lung disorder called pulmonary alveolar prostenosis may develop, which is when excess protein builds up in the alveolar sacs, making the ability to breathe very difficult. LPI is diagnosed after urine samples are taken over a 24-hour period, and the results show an output of the 3 amino acids that are not being digested. So to be frank, instead of starting from the top of the list of case studies, I went to the very bottom of the list and worked backwards until I found something that looked intriguing to me. I have never heard of Lysinuric protein intolerance, and after some surface-level research, I found that I wanted to learn more. And having a 5-month-old myself, I could not imagine having my child diagnosed with such a disorder after weaning off breast milk, especially after they seemed perfectly healthy just prior to. So, founder effect is when a small group of a larger population leaves and starts their own colony somewhere else. This has a great genetic disadvantage because a small colony now has a greatly reduced genetic variation than a larger population from where they descended. A popular example of the founder effect is a polydactyl mutation within the Amish community. The polydactyl trait, which was once a rare occurrence within the larger population from which the Amish descended from, is now very common because the mutation within the new and small genetic pool lacks genetic diversity. If the founder effect were to take place with Lysinuric protein intolerance mutation, I cannot imagine that the colony would survive for many generations due to the numerous children that would be passing. The founder effect is also why LPI is prevalent in Finnish and Italian families. So as mentioned previously, consanguinity is a situation in which a couple who plan to procreate are related by blood. Consanguinity and the founder effect go hand in hand here. Consanguinity can be a common occurrence within a small, isolated population that has left a larger population, like the Amish community described earlier. With less genetic diversity and the lack of diversity within the populace, it is more common to see blood relatives procreating, thus exponentially increasing the rare disorder's occurrence, causing it to become common rather than rare. And LPI is a autosomal recessive disorder, and currently there is no treatment for LPI, only symptomatic treatment, which can be very extensive and involved, thus requiring specialized medical professionals and facilities to care for the patient. And because the location of the mutated gene is known, perhaps someday recombinant DNA can be produced containing an unmutated gene, and it could replace the damaged DNA before cell differentiation. So a mutation such as this could occur from a splice site mutation. In eukaryotic DNA, coding regions called exons, which are interrupted by non-coding regions called introns. In order for the primary RNA molecules to be matured, it requires the removal of the introns. However, sometimes this is not a clean cut, and some of the exon is removed also, causing a mutation within the final DNA. So, in relation to our course and our studies throughout these 8 weeks, we have gone over many different types of mutations, and LPI in itself is caused by a mutation of the SLC7A7 gene, which is located on the 14th chromosome. And if you would like to learn more about Lysinuric protein intolerance, please refer to these sources. Thank you.